Right, in today's lab, we are going to explore how enzymes function over time and in various environmental conditions. So as we might remember, enzymes are biological catalysts. That means that they are going to lower the amount of energy input, right, that activation energy that is required for the existing bonds to become unstable so that the reaction can occur. Here's my classic drawing of an enzyme. And remember that enzymes are typically proteins, with one weirdo example that we'll talk about later. We don't need to talk about it right now. Okay. And each enzyme, the shape of that protein is going to determine if that enzyme can do its job. Okay. And each enzyme is going to be specific to a substrate. Okay, that's the molecule that this enzyme is going to work upon. In our case, for today's lab, that substrate that we're going to be working upon is called lactose. Okay, this is a disaccharide, a carbohydrate. It's often found in milk. It's known as milk sugar. So if you happen to be lactose intolerant, you lack the enzyme of lactase. Okay, and most enzymes have the ending of "ase" at the end of their names, which is convenient. And so what lactase is going to do is take lactose from its dimer state of a glucose molecule and a galactose molecule, and it is going to catalyze a catabolic reaction that is going to break that disaccharide into two monosaccharides. So what we're going to get out of this after this reaction has occurred is something like this, where we have a galactose as a product, right? That monomer, that monosaccharide, and we have glucose as the other product. The way that we are going to measure how well lactase is doing its job is we are going to be measuring how much glucose, right, the concentration of one of the products, as how well this is going. So in our initial solution, we should expect to find no glucose because it's all bound up with the lactose. But once we have the enzymes be able to break that apart, then we should start to be able to detect that.